My name is Jane Fife, and I'm here to educate today. I work in the biologicals industry for a company called 3-Bar Biologics, and I've been coming to this conference, this Conservation Tillage Conference, for probably about 6 or 7 years now. When I started working at 3-Bar Biologics, and in the early days, we're a startup, a company based in Columbus, Ohio. In those early days, we had a booth down in the cafeteria area. We brought some of our products that we were developing and got questions. I think there was curiosity 6 or 7 years ago on what we were doing. We have a new kind of delivery technology that we were developing at the time, and that curiosity was coupled with a lot of open skepticism on these biological products. And some of those conversations were a little rough. And so every year after CTC, at some point I would run into Randall Reeder, and I saw him running around in the last session. And I'd say, hey, Randall, biologicals, anytime you want me to come talk about them, I'm happy to do so. Well, after 7 years, here I am, right? But what we're seeing in the industry, certainly, and Laura talked about it, this increase in interest at Commodity Classic. We've been going to Commodity Classic for a number of years. And the number of companies that keep showing up increases, the number of products keep increasing. And for you guys, that's challenging, right? You're kind of getting inundated with all this stuff, and you don't know what to do with it. So my goal today, being here, is really to educate, to inform, and hopefully you guys will leave with some learning outcomes. And so I've broken the presentation up into kind of 3 segments. I know biologicals are challenging to understand, so we're going to start with just what are they. There's a range of different market segments and product categories. I'll get into that a little bit so you understand what they are. How to apply them. At 3-Bar, we are very focused on the delivery. It matters what you receive at the farm gate that those microbes are living, and how they're delivered matters just as much as how they were manufactured. So I'm going to talk about delivery technologies, some of the things we've learned working with partners. And then there's a lot of field trial data out there, and I think what I appreciate seeing now, I certainly appreciate our extension folks working on creating more data here in the US. But there's a lot of data that we're now starting to see what's called meta-analyses, where they're looking across all of that field data and trying to get more insights. And so I'll share some of that information. When Stephanie said I could have an hour, that's dangerous. So I think I added too much to this presentation. So there might be a point where I'll have to I want to make sure there's enough time for questions. So we'll see how this goes. But there's a lot here to unpack, and I'm sure there'll be questions. I'm actually going to present again, not just once today, twice, at 1 o'clock or 1:30, somewhere in that time frame in the chapel. And that's going to be a little bit more. This is really focused on biological seed treatment to kind of build on what Laura just presented. The afternoon presentation will be looking at biologicals more generally. Okay, so here we go. All right, I know there's a lot up here, but I like this diagram because it starts to really get organized around what biologicals are. And so biologicals are, get my pointer going here. It's not working, there we go. All right, biologicals they include biological control products, biostimulants, and biofertilizers. Those are the main market segments, and that's, as an industry, how we start to market these products, right? Within, I think biocontrol products are easy to understand because they kill or deter a pest or a pathogen, right? And there's been biocontrol products on the market for a long time. A good example is Bacillus thuringiensis, or BT, which is a commonly used bioinsecticide. But there are macroorganisms, microorganisms, and biochemicals that all fall under biocontrol, okay? The thing with biocontrol is that it is regulated by law under FIFRA. So these products go through a very rigorous evaluation before they make it to the market around safety and efficacy and so forth. So there's a regulatory framework in place. When we talk about biostimulants and biofertilizers, right, so a lot of these biological seed treatments that we're talking about, currently, there's no regulatory framework in place. So what happens when products aren't being regulated, right? The bar is low. You have a bunch of products on the market that aren't really being evaluated at the level that they need to be. And so what do we see? And I know you guys love this word. Begins with an S. Snake oil. Some of you like foo-foo dust. But these are products that don't work. And they don't work because, one, there may be no living microorganisms in them, which is a problem, right? They haven't been adequately tested, right? And so this has put a big black eye on our industry, okay? For a number of years, our industry, working with government partners and officials, have been working hard, both at the state level and the federal level, to get regulatory framework together. And so after a lot of years of hard work, the first major outcome of this is a bipartisan effort that came out last year with the Plant Biostimulant Act. And what the Plant Biostimulant Act will do is it will create biostimulants as its own product category and establish a regulatory framework around that. So by law, we will start to see regulation happening. These products will still be registered at a state level, but there'll be a standard definition, which I'll show on the next slide, and more uniform labeling, which will make it easier for industry and for you all. But the point here is these two market segments around biostimulants and biofertilizers, which tend to be associated with plant health and not biocontrol, will have a higher bar, right? And we'll see that there's more and more products coming on the market, like we've discussed, but we'll start to see, the technology is improving. We're seeing better formulations. We're seeing better microbes. And so you're going to see that happen, especially as that bar raises with these new regulations in place. Okay? So good news. The Plant Biostimulant Act, by definition, like I said, establishes plant biostimulants as a product category. The key words here, this means that these are either a substance, a microorganism, or a mixture thereof. I like to think of the substance as the dead, right? So this is, for a biological product, this is anything that's been derived from typically a biological source, right? It's naturally derived. So it's bioactive compounds. It's something that's either coming from a microorganism or a plant, but it's dead, right? It's just those bioactive compounds, okay? A microorganism is the living. So it is a living organism. And in order for it to provide a benefit to the plant for plant health purposes, the microorganism needs to reproduce, associate with the plant, and be alive. There's a lot of crosstalk that happens, and so this is important that it's living and viable when it's delivered, okay? So keep that in mind as we talk more about biostimulants, kind of the dead and the living. And so they're applied to seeds, plants, the rhizosphere, soil, or other growth media, acting to support a plant's natural processes independently of the biostimulant's nutrient content, right? It's not a fertilizer, right? Including improving nutrient availability, uptake or use efficiency, tolerance to abiotic stress, this is a big one, and consequent growth, development, quality, or yield, okay? That's the definition that took, I'm telling you, years to come up with, but it's here. And we're gonna work towards a regulatory framework. All right, I'm gonna focus today's talk on microbials, okay? Biological seed treatments are typically associated with microbials, so microbes. You can see that microbes occupy both biostimulants. This is the living part, microbes. There's also other product categories that represent the dead, the substances, but I'm going to focus on the microbes here. And then biofertilizers are all microbials. Okay, so when we talk about nitrogen, biological nitrogen fixation, it's a microbe that's doing that. When we talk about phosphorus solubilization or mineralization, it's a microbe that does that. Okay, and typically we have bacteria and fungi that are the main product categories involved with as either biostimulants or biofertilizers when we're talking about microbes. And the bacteria, the researchy term that's been around for a while, is plant growth promoting rhizobacteria or bacteria, PGPRs. So you'll hear that term, PGPRs, used. You'll also hear the term inoculant used, which inoculants are kind of a carryover to soybean inoculants, right, where we're inoculating the seed with a microbe. Okay, so those are two common terms used particularly for bacteria. And what PGPRs or plant growth promoting bacteria do is, naturally in the soil, there's plenty of bacteria and fungi. And the plant actually draws to it the microbes it wants through this very complex signaling that happens. The plant is excreting in its root system root exudates that kind of feed the microbes, and they're attracting certain microbes. So there's this signaling that happens, right? When we introduce a microbe product with the seed at planting, what we're doing is we are kind of accelerating that process with specific microbes that have specific functions that we know will benefit the crop and help either through abiotic stress or help it with nutrient uptake and use. So you have both the ability to fix nitrogen. Some microbes, not all, do that. Solubilize nutrients, in particular phosphorus. And then this other aspect, plant phytohormones. This is key to biostimulants, where there's certain compounds that the microbes produce that can help the plant through different stress responses or help them more vigorously grow their root system. So there's certain phytohormones like auxin, gibberellins, cytokinins that are known to do that and have that effect, okay? So knowing what the microbe does is very important, okay? And when I get, later I'm going to talk about how to understand a label and reading a label to understand what the microbes are that are there and what they do. It's really important to these products. I think Laura said, these microbes do different things, and understanding what those different things are is really, really important, okay? To mention fungi, the common class of fungi are these arbuscular mycorrhizal fungi, or AMF. Their function, fungi will grow and colonize a root system and kind of provide these extensions of the root system through the hyphae that they produce. So it allows that root system to reach wider, which helps with water and nutrient uptake. So it has a very specific function in helping with plant health and water and nutrient uptake. So those are the two broad kind of categories between bacteria and fungi that we see as microbial products that are currently on the market. What's going on? The way that I like to think about beneficial microbes is they're basically little chemistry factories. They're living organisms like us. They like to eat. So when they intake food, which tends to be carbohydrates or sugar, amino acids, organic acids, they will then produce biomolecules. And those biomolecules will range. It's very complex. There's a range of different molecules that these microbes produce. What the microbes produce depends on their genetic makeup. So we know that we've talked about genus species, different genus species will have different genetic makeups, and depending on the conditions they're exposed to, certain genes will be upregulated or downregulated, and we'll see different biomolecules produced. So it does become very complex fast. The key here is that they need food, and then they're going to start to produce this range of chemistries. What makes this such an interesting system when it works is we can't produce the complexity of this chemistry in a lab, right? We can't synthetically make these chemistries. But what makes this so cool is that when that signaling is happening with the plant, it's an active system. It's an active biological system where the chemistry that these microbes are producing is a reaction to the plant. So if the plant's stressed, the chemistry that these microbes are going to produce as beneficial microbes will help the plant through that stress. So it's a very dynamic and active process, which makes a living biostimulant or biofertilizer product different than a dead product, if that makes sense. Okay. Oh, all right, I jumped ahead here. So Laura did a great job talking about the disease triangle, and I think it's worth repeating. I do the same thing when I present because I think this mindset we're all familiar and comfortable thinking about the disease triangle and the fact that we don't want a disease to take off, right? There's certain conditions where we'll see a pathogen take off. We have to start to kind of think about this the same way with inoculants. So I like to call it the inoculant triangle, right? Where we do have to make sure that we have a welcoming host. So a lot of attention in the cultivars we're using, the right microbes with the right cultivars. You'll see more research in this area as we're really just at the beginning. But making sure there's a good match there. And then having a favorable environment. Some of this we can't control, but some of it we can in terms of the inputs that we're stacking or delivering the microbes with, okay? So we do have an opportunity to, at a certain level, to control that environment. And then the microbe itself, making sure that it's highly viable, very fit to be able to survive and proliferate in that environment, right, with the seed at planting. All right, so I'm going to move on to application and delivery, because this is kind of where the rubber hits the road and where a lot of these products, quite honestly, fail, right? It's how we deliver them. And if anything, pay attention to the next minute or so, okay? Two key things I want you guys to take away. When we talk microbials, there's two things, CFUs and logs, okay, that you have to keep in mind. And what a CFU is, it's a colony forming unit, is what it stands for. And it's the way that we measure viability. We can quantify in the lab viability of these microbes. So if I take a sample out of a product and I plate it, we use these plates, we spread plate the sample, I'll be able to, the microbes should grow. If it's pure, you'll see just one colony there, type of colony. But then I can count them, and so that's what this is. It's a way to quantify the number of viable cells that are there, okay? The other thing to keep in mind is logs. So anything microbial is on an exponential scale, okay? So it's kind of hard to get from a linear scale to an exponential scale, but in microbiology, everything's exponential, so we talk in terms of logs. And so what this means is, if we start with 1 million cells, okay, this is 6 logs, so 10 to the 6, okay? Thing to keep in mind here is if I lose a log loss, 1 log, that means I'm going from 1 million cells down to 100,000, okay? So I'm going from 6 logs to 5 logs, okay? That log translates to 90% of the microbes. I've just lost 90% of my product, right? If we were talking about this in chemistry terms, that's horrible, right? Your dose rate's, out the window. In microbiology, this is, log loss is a big deal, and we see log loss seriously in every step of the process, and that's where we're trying to improve our practices and make sure we don't see log loss throughout the application process, okay? And as you go kind of down in terms of more log reduction, you get to 2 or 3 log loss, you're talking 99%, 99.9%, you're losing most of your efficacy right there, okay? So keep that in mind, CFUs and logs, okay? We're going to talk more about that in the coming slides. All right, it's important to understand, particularly with the biological seed treatment, the number of CFUs per seed is really important, okay? There's various data out there. This comes from a study from about 10 years ago where they at least started to look at different countries and where there were regulations versus where they're not. And I will say in 10 years, we haven't gotten very far. Regulation varies widely. In the US, there remains no regulation or requirement for number of CFUs per seed. But the general consensus from industry and from regulators is that particularly for corn and soybeans, so larger seed sizes, there should be at least 5 or 6 logs. So 5 or 6, 10 to the 5th or 10 to the 6th CFUs per seed. Okay, so 100,000 to 1 million bacteria cells or CFUs per seed. Okay, so that's the general standard we're trying to meet. And like I said, in the US, there's currently no standard in place, right? But that's what we're trying to achieve. Okay? So keep that in mind. All right, understand the label. Okay, this is actually a product that was in the study that Laura just talked about. And one of the first things you'll want to look at the label is understand what microbes are in there. And so you'll see the genus and the species, so a Bradyrhizobium alkani, and then a Delphtia acidioborans, so 2 different microbe strains. You'll also want to note how many CFUs per mil. For a liquid formulation, you talk in terms of milliliters, so numbers of CFUs per milliliter. Or if it's a dry formulation, it'll be CFUs per gram. So you want to kind of understand where you are with that concentration. And so in this case, we're at a very high concentration for the Bradyrhizobium at 10 to the 10 CFUs per mil. For the second strain in there, we're at a much lower level, 10 to the 7th CFUs per mil. So quite a bit lower. And I'll talk about that here in a second. Understand how it works. The Bradyrhizobium, I think everyone understands, these are Bradyrhizobium, Rhizobium species are specific for soybean inoculation for biological nitrogen fixation, right? There's a history with these strains. But what does Delphtia do? I've never heard of Delphtia, right? And so a good product label will include an indication of what all the strains are and what their function is and what their purpose is. Not all labels currently include this information at a good level, but some do. In this case, I know it's hard to see, but they do indicate that Delphtia acidioborans is a plant growth promoting rhizobacteria, APGPR, okay, to enhance plant growth and yield. So it's there as a biostimulant to help with stress response. The label doesn't tell you exactly what it does, but it's there to help with growth promotion as a PGPR, okay? So that's good. We have an understanding of what the strains are and what they're there for. The next key bit of information is your application rate, okay? And this application rate between your minimum guaranteed analysis of what's there in terms of CFUs, you can then calculate what the application rate should be on a CFU per seed level based on using the application rate there, rather, to calculate, okay? And so in the case of the Bradyrhizobium, because it is at such a high concentration, 10 to the 10 CFUs per mil, the application rate's essentially 22 mils per 140,000 seed. We see through calculation, we get to about 10 to the 6 CFUs per seed. So that aligns well with the standard, this general rule of thumb of being in the range of 10 to the 5 to 10 to the 6 CFUs per seed. Okay, so that sounds good. Now the second strain, the Delphtia, when we do the same math, we arrive at roughly 3 times 10 to the 3rd, so 10 to the 3 logs CFUs per seed. A lot lower, right? And so you're talking 1,000 cells or 1,000 CFUs on each seed is what this formulation's providing. Maybe that's enough. I don't know. I didn't do the trials on this particular product. But to me, that's a low number of cells. If we're trying to achieve 10 to the 5 or 10 to the 6, 10 to the 3 doesn't sound like a lot. Something to look into and better understand, okay? Things to look at. If there's a low number of low concentration of cells to begin with, I would question what they're there doing, okay? So understand the label. And the last piece here is there's usually indication of how to store the product, how to apply it. Make sure you read that on the label. Most biological products that include microbes, they either need to be stored at room temperature, so 70 degrees, typical room temperature, or be refrigerated. If the product needs refrigerated, it should say in bold letters it needs refrigerated, and do that because those microbes aren't stable at room temperature, and there's still quite a few products on the market that need refrigeration. But the ones at room temperature should be okay for the shelf life that's indicated on the product. If they are exposed to elevated temperatures, so above room temperature, we're talking 80, 90, 100 degrees Fahrenheit, right? Hot temperatures. If they're in the back of your pickup truck, in the sun, probably not a good idea, right? These microbes are going to get hot and die, basically. So you're going to get lots of log losses if you leave the product in not right conditions, okay? Temperature's a big factor with these products, right? So either room temperature or refrigerated temperatures tend to be okay for storage. The other thing to keep in mind is take the product, before you actually use it, you should give it a good, if it's a liquid formulation, you should give it a good shake, because things settle out. These microbes, depending on what the formulation is, you just want to make sure that you give it a good chance to be suspended in when you actually use the product. So those are just a couple key things to keep in mind. All right. The importance of viability. Okay, I talked about log loss and the challenges with these products, right? You want to have viable, high concentration of viable cells on arrival, period. And the reason for this is you're basically taking these microbes and putting them in an environment in the soil with the seed at planting, okay? And what happens is these microbes, they're going to experience competitors, so there's other microbes that are in the soil that they're going to have to compete against for resources. The conditions, temperature and moisture, pH, can matter with these microbes. And then time, because what's happening is time before the seed germinates, right? And the seed is, as that plant root system, the seed starts to germinate and the root system starts to emerge, that's food for the microbes. And absent of that, the microbes are just kind of hanging out. And what happens with time is you're going to start to have log loss, because if they don't have food, they're going to start to die, okay? If there's a little bit of moisture there, they're alive, and if they're not being fed, you're going to start to see some log loss. So even if you delivered a product that had a high concentration, you may lose some during that delivery process before the seed germinates and is actively feeding the microbes. There may be nutrients there in the soil solution the microbes can snack on, and that's why what you deliver them with can really matter and help create a better environment for the microbes, okay? What you want is to have a high population there that survives, right? Enough there that survives. And again, going back to the label, if you're starting with not a lot and you lose some, you're left with close to nothing, right? Which is why sometimes products aren't working in the field, right? But what you want to happen is have enough survive, and then they're going to start to colonize the root system. So this proliferation happens. The microbes are being fed. They're producing this range of wonderful biochemistry that is signaling to the plant, and you've established this nice biological system with this input application, okay? That's what you want to have happen. What root colonization looks like, it's basically those microbes growing down the root system. In this case, this is just a tap root. You can see as that root started to germinate, you can see in this case a Pseudomonas species. Pseudomonas are known to be very good root colonizers. There's a number of reasons why they're very good, but you can see that it continued to grow with the root based on this assay. Okay, that's what you want to happen. All right, so how we deliver matters, okay? With biologicals, just like with chemistry, right? We are focused on precision delivery, right? And that's why seed treatment is convenient. It's nice and convenient, right? We can put a lot on the seed, but there's only so much room on the seed, right? And what's there can impact, if you add a microbe, it can certainly impact the microbe. Liquid versus dry for seed treatment is important. And I say this because liquid seed treatment is convenient, and it's how mostly it's being done. But what happens to the microbe, it's a liquid, it has to dry, and the desiccation process that the microbes experience during seed treatment actually contributes to a lot of loss of the microbes, because they're going through this very uncontrolled drying process that happens during seed treatment, and you start to lose some microbes. So that can be something you have to be mindful of, the seed treatment process, and how many microbes you might be losing through that process. With a dry formulation, dry powders are not fun to work with, I know, but the microbes in a dry formulation, in a controlled, high technology way, have been put into this dormant state. So they actually have a better shelf life than in a liquid formulation, and they're just kind of hanging out. And so when they get applied to the seed as a dry, as long as the moisture, you can maintain them in that dry state, they're going to do better over time and not have to go through that cycle of what I call the cycle of death, where you're trying to dry them down in the seed treatment. So you would, in essence, in theory, lose less with a dry application, okay? So there's a difference between liquid and dry. Seed treatment, obviously, like I said, is convenient. Liquid in furrow is another way to deliver biologicals. I know not everybody has in furrow equipment, so there's limitations there. But the nice thing about a liquid in furrow is, you know, those decisions, you have flexibility. You do have to add the microbes into a tank mix, so what's there in the tank mix matters. I'll share some data. So there is the potential for, you know, you want to make sure your microbes are compatible with what's in that tank mix, if it's a starter fertilizer, any fungicides or insecticides. But that's another common way to deliver them that kind of overcomes the issues of the dry down on the seed and any loss you might see there, okay? 2x2, I get asked a lot about delivery of microbes by 2x2. I wouldn't do it. The fact of the matter is, when they're that far away from the seed, they're waiting. And if there's no food there, you're going to continue to see loss of the microbes. Other biologicals that are not living, right, the dead, 2x2 might be a great option, right? There are substances, and depending on what they do, 2x2 is used with some biologicals. I just wouldn't recommend it for microbials, for obvious reasons. Foliar. Foliar is kind of what I'd say an up-and-coming way to deliver some biologicals, not all. And I say this because there are some interesting new products out there in the nitrogen fixers that they're a type of microbe that's called an endophyte, and that means that they get into the plant cells and can replicate and fix nitrogen from the atmosphere. And as a foliar application, we're seeing really good success, particularly for corn, particularly later in the season, right at that V4, V6 when the plant gets really hungry. So foliar is being used a little bit more for microbes in the biostimulant and biofertilizer space. So we'll start to, I'd keep an eye on that as we learn more about those foliar applications. But the downside of a foliar application is your spraying requires more material. It's not as precision delivery as you would with the seed or as an in-furrow delivery. But something to keep an eye on. All right, the next couple of slides, I'm going to go through some equipment. This is probably old news for some of you, but I'll go through it really quickly through the lens of applying a microbe product, okay? And some things to keep in mind. So when we are dealing with like a bulk seed treatment, you know, I've talked about this impact of dry down, the desiccation loss that microbes will experience. Most seed treaters, the big ones, have an inoculant port built in, and this was an adaptation made for soybean inoculants. And the idea is that all your chemistry is being applied through the atomizer from a separate tank, but that you have a port where the inoculant, the microbes are being kind of dribbled in, okay? Separate. So you can keep them separate. You don't have to tank mix them with the chemistry, and they're being dribbled in after the chemistry is applied. So that's definitely a move in the right direction to keep the microbes separate from the chemistry and not put them through an atomizer. But they're still going to have to dry down on the seed like I've been talking about. For distribution on the seed, again, getting back to this idea that we can have 10 to the 5th or 10 to the 6th CFUs per seed. You want to make sure that you have good distribution of what you're applying so that you're getting to those levels on your seed. And a lot of that happens with bulk seed treatment through seed tenders, conveyors, right, where that transfer of seed, the mixing that's happening is allowing the seeds to touch each other and fully mix, okay? So that's important, particularly when you're trying to achieve these levels of CFUs per seed, okay? With dry, oh my goodness. There's so much room for innovation in applying dry inoculants. Where we are today for applying dry, we have equipment being used typically for seed lubricants, right? So talc, graphite, and others. And that is actually a really good vehicle if you add the microbe inoculant with the talc graphite or the seed lubricant. Add them together and then deliver them, right? Challenge is, you know, we're trying to get a dry powder onto seed. So I think we'll see innovation in this space, but it is a challenge. But a good way, like I said, if the microbes dry, you're not having to experience that same dry down loss that you are with a liquid seed treatment, okay? And again, you know, the seed tender and the conveyance and transfer helps continue to distribute the microbes onto more seed. Newer seed treatment technologies, you know, pro boxes are very popular, and so the ability at a retail or farm level to treat seed from box to box, you're seeing some new technology. Thing to keep in mind in terms of biologicals, you know, whether it's a nozzle in the chute as the seed comes out and being transferred from box to box, or an auger type of system, a little bit bigger system here, you're still using an atomizer. There's no separate exit point for the microbes, so you have to keep that in mind. And there's more limited conveyance here, so that mixing, making sure that the distribution's adequate is something to keep in mind too. But these are newer technologies on the market, and how you fit microbes into it needs to be considered if you're using inoculants with chemical seed treatment. And the last one here, in terms of seed treatment, is what can happen, you know, in a planter box or a center fill. This is, you know, an option you have direct on farm. You don't have to deal with anything upstream. And so again, it comes back to a dry. And so the high-tech way we do this, unless you guys tell me something different, I might learn today, is sprinkle and mix, right? You take a little bit of the dry, you sprinkle it in, and you mix it with something, right? And that's kind of where we are technology-wise. But again, remember that you need to have it fairly well distributed, but getting that dry on the seed at the planter box. There's quite a few products on the market that's how they've gone to market, is using this form of application, okay? Again, using seed lubricant as a delivery vehicle. How am I doing on time? I don't think that clock's correct. We're good? Okay, I'm marching through these, so maybe I will get through them all. All right. The last piece here gets to liquid and furrow around application, and I'd say there's room for innovation in dry application. Where we've seen a lot of innovation is with liquid and furrow. And there's been a number of new technologies that have made it to the market recently. This isn't an exhaustive list, but a couple to mention here. Precision Planting has their new furrow jet. The nice thing here is you have multiple streams, so the ability to separate out the biological and have it be its own stream is doable with this system, so that's nice, especially when you're thinking about tank mixes and trying to protect the microbe from the chemistry you're applying. Another technology that's very recent out is 360 Yield's 360 Dash. And in this case, it is one stream, but it has an on-off valve so that where you don't have seed, it's off. So you're dashing basically the liquid to where the seed is. So it can help you reduce your application rate, which may be nice, particularly with more expensive products, right? So another nice innovation in furrow technology. And then this other one, this is actually a startup, Inflection Point Technologies is a startup based in Iowa that I don't think their product's on the market yet. They're still in development. But it is an on-the-go seed treatment. So you're basically spraying the liquid onto the seed as it's going down into the furrow, which again makes for a very nice system for microbes, so that dry down isn't happening the same way as a upstream seed treatment. You're getting the material on the seed and it's going right into the soil, okay? So new technologies on the horizon that I think favor microbe products very nicely, and something to keep an eye on. All right, now the downside, compatibility. I talked a lot about the stress that microbes get during dry down, that desiccation stress, which can kill microbes. The other main thing is compatibility with chemistry. So whether chemistry has a toxic effect, depending on what the chemistry is, or an osmotic effect due to salt, or mainly salt. That can all impact the viability of the cells and either leave them in a not good state or completely kill them. So this is where you can see a lot of log loss. The thing to keep in mind we'll start with fertilizer and salt index. When you are tank mixing the microbes, particularly with if you're doing a liquid in furrow and you're tank mixing with starter fertilizer, for the seed's sake, typically you're going to use a lower salt, that's what a starter is, right? Lower salt index. But a lot of growers use 1034-0. A lot of you do. I know this. And 1034-0 is not good for microbes in general. Some are okay, but in general, no good. And so 20 is kind of this break-off point. And what we typically recommend is something a good bit more milder, which is a 624-6. And we've seen pretty good compatibility across a wide range of microbes using a 624-6, so something with a much lower salt index than 1034-0. The other thing to keep in mind is the pH. So the pH of the solution you're delivering with, if it's a liquid, but also consider the soil. And most microbes don't like low pH. So you get into the range of 4, 4.5. That's not so good. 5, sometimes not good. More neutral 6, 7 pHs tend to be just fine. Okay, so keep that in mind, pH. And then, depending on what the nutrients are, any micronutrients, there are some micronutrients that are great biocides. Copper is a great example. It kills bacteria. So if you're using copper as a micronutrient, just be mindful of that. It may not be compatible with your biological. So checking compatibility. Most manufacturers are already doing this. We do a lot of this work in our lab at 3Bar. I'm going to show you just some quick data here. This comes from last year's field trials where we had a Pantoa strain, actually 3 different strains that we were evaluating in field trials. And so we had 4, these were on production acres. We had 4 different growers involved in the study. And we asked them ahead of the season to send us samples of their tank mixes, because this was going to be delivered in furrow. And we created these great little kits. They had these little vials, labeled, bags. It was just this nice little kit to send it to us back, really organized. And so this is what we got back from one of our growers, the starter fertilizer in a Coke bottle. And my lab manager, there was steam coming out of her ears. I thought it was kind of funny. I know y'all are busy during that time of year, and maybe the Coke bottle was just easier than digging out the nice kit that I sent, but I thought it was funny. In any case, what we found, and so the colors aren't coming through real great here, but these are all log losses, okay? This is 3 different strains of this Pantoa bacteria that we're evaluating in field trials. Everything's being controlled compared to a buffer control where there's, that's our control that we're comparing to in terms of CFUs, what's there. And so after sitting with the tank mix for 6 hours, what did we have left? And so these are the log loss of what we lost based on what was there to start with. And you can see anywhere where there's 1 or greater or even approaching 1, this is a problem, okay? So if you remember when we talked about logs and log loss, this means we've lost just from sitting with the starter for a little bit over a log, right, or a log. So that's 90% of the microbes gone, right? So something to keep in mind. 1 log doesn't sound bad, but 90% sure does, right? And so when we took the 1034-0, and that's what I said, I mean, it varies from strain to strain. You can see these differences, but in general, it's not good. But one of the tricks, you can dilute it in half. That might be okay. In our case, at least for it sitting together for 6 hours, we didn't see much of an impact. See these low numbers here. So diluting the 1034-0 can help. The 624-6, like I said, we recommend it because it is gentler. Again, we saw good results with it. This one, I hope you're seeing this one. This is water. We had one of the growers send us their water. Think about it. Be mindful of your water source. So whether it's city water or from a well, city water often has chlorine in it. That chlorine can fluctuate and vary. Chlorine's a great biocide. It can kill microbes. That's why it's there. Minerals. Sometimes from well water, you have minerals that can impact the microbes as well. Okay? So this was a surprise to us and something I'd just like to point out. You do need to understand what you're mixing with, even the water. And then we let it sit for 24 hours, and you can see that where it was bad, it got worse in terms of log loss. And in some cases, something that sits for, like this Ignite was a product, it's basically an extract that has carbon and amino acids in it, so a carbon-nitrogen source. It was okay for the most part. It did show a little bit of sensitivity of one of the strains. But then after 24 hours, multiple logs lost. The microbes didn't like it at all for whatever reason. And so something to keep in mind is when you tank mix and you put microbes in it, use it the same day, right? 6 hours is a great, do it in the morning. By the time you get to the end of afternoon, you've used most of your tank mix. Microbes, as long as they're compatible, will be okay. But leaving it a day or more is not good. You should use that tank mix the same day. Okay. So log loss on seed, again, this dry down effect I've been talking about. There's not a lot of data out there. I don't have any from my lab that I can share. But there was a nice article that came out this year. This is out of Brazil, looking at the impact of chemistry on seed with, in this case, Bradyrhizobium strains. And what they found is you can see after 3 hours the impact, which is not much, but then as it sits for days, this is without chemistry there. The impact, you have log loss, multiple log loss happening the longer those microbes sit on the seed. And when you add chemistry, these are different chemistry packages, fungicide and insecticides. You can see that that impact is worse. In some cases, it happens right away, where there's several log loss happening compared to the control. So be mindful of that. You're going to have log loss on seed. And the further upstream your seed's treated, the more log loss that's happening over time. And it depends on what it's being treated with as well, okay? All right, the last piece here gets into the field trials, and this is where I got nervous on time. I know I don't have a ton of time left, and I want to make sure there's room for questions and stuff. So I'm going to go through this quickly, kind of, hopefully. But one of the things I wanted to point out, this is a survey that came out last year, McKinsey Group, where they did a global survey of adoption of biological products, and they did a nice job in terms of breaking it up into the different market segments of biocontrols, biostimulants, and biofertilizers. And what they found, I know this is hard to see, this is Brazil up here. In Brazil, the adoption is very, very high compared to the US and Canada down here. So these are currently planning to use the dark blue and the lighter blue. So it begs the question of why, okay? And we talked a little bit, Laura mentioned this in her presentation on, Brazil is actively using microbial inoculants. And I'd like to point out 3 things, because I think it's important here, on why Brazil has adopted biologicals. They started out doing very extensive research over decades with certain soybean cultivars. This is all really focused on soybean to start. And they found the elite strains that did best with those soybean cultivars. And so they have 4 that are used extensively within Brazil. So a lot of this upfront work targeted at certain cultivars, certain conditions, okay, to come up with these elite strains. The other thing they did, they established law that said there are certain commercial standards that have to be in place, right? They have a minimum concentration of 10 to the 9 CFUs per mil. If you're going to have a commercial product in with these elite strains in Brazil, it has to meet this commercial standard. Very important, okay? And they have strict quality control. So they actually monitor what's coming in, not only products that are produced in Brazil, but ones that are imported into Brazil, okay? This is a recipe for success, right? And I talked at the beginning about the importance of having a regulatory framework. And we're going to see that success catch up in the US, right? But I think this is true to why these products are being used so extensively in Brazil, okay? The next few slides I'm going to go through pretty quickly. And there's a lot of data, and your eyes are just going to glaze over. But I'm going to point out a couple key things. And so these data come from work out of Brazil, where they've looked at all of this extensive research, field trials done over several decades, lots of data, and they've done these what's called meta-analyses. Okay, so they're looking across all these field trials from different regions of Brazil, different products, and they're saying, okay, what insights can we gain from all of this work? And so those insights end up being the effect. And in this case, what they've done, building on their elite Brady rhizobium strains, they're co-inoculating now the Brady rhizobium in soybean, which we know Brady rhizobium fixes nitrogen in soybean, with an Azospirillum brasiliense species. And the important thing about Azospirillum, it's a free-living bacteria. It fixes nitrogen, not as efficiently as Brady rhizobium does with soybean, but it also is a PGPR. So it produces a range of hormones, plant hormones, particularly auxin, which supercharges root growth. So it does have a plant health benefit. So we're going to co-inoculate with both of these. So multiple functions happening here, okay? And what I want you to notice is anywhere where there's data points that are skewed a little bit this way is where we're seeing a greater effect, okay? So boom, where do we see the biggest effects? When we look at how they were applied, we see in furrow did a little better than on seed treatment and foliar. We see a dry seed treatment did a little better than a liquid seed treatment. Furrow was right there behind it. So you see some of these trends starting to merge, these insights from these meta-analyses. Lower yielding, we talked about that. When your yield is a little lower, there's a chance for a better impact of applying these microbes, right? Sandier soil, so we start to look at sandier soil, which we see more in a drier climate and even in tropical climates. So not as good soil as we have in Ohio, Indiana, Illinois, Iowa, right? We're seeing a much bigger effect than other better soil conditions. No-till actually has a slight advantage over conventional, and I think here we're starting to see the impact of more soil organic matter in there to help the microbes be a better environment for the microbes, right? So no-till can have an impact. And then drought, when there's not enough water there, and Azospirillum, because of its ability to help supercharge that root growth, is helping plants through if they have challenges with droughty conditions, okay? And then another study they did now, looking at, we tried Azospirillum in soybean, now let's try it in maize. So again, out of Brazil, looking at the use of Azospirillum, which can fix nitrogen, it's not as efficient, but also can produce plant health benefits. And so you see here, boom, root mass, which is what you would expect to see a big response in additional biomass in the root system. Again, dry seed application being used saw better overall impact than liquid. If I go to the next slide, boom, this is the same study. They saw as directionally as soil organic matter increased, there was a greater impact at higher soil organic matter. This goes along with a little slight advantage of no-till practices, contributing to the higher soil organic matter. Sand again, sandier soils, they saw greater impact, lower yielding, saw much greater impacts. And then here's a key point here, where the soil nitrogen was lower, you saw more of an effect, right? So again, going back to Azospirillum being able to fix nitrogen from the atmosphere versus when it's all, there's a lot there. All right, the last slide here with meta-analysis, I think ties to some of the observations that Laura was talking about. This is a different study, not based in Brazil, but where they're looking at trial results across globally. So all these little points over here represent different climate regions. And so the red dots, which are hard to see, I know, represent a dry climate. And you can see through southern US and over into Europe, and a tropical climate, which is the blue dots, Brazil and Southeast Asia. This is where we see the biggest impacts of using these biofertilizers, either a dry climate being the most or tropical climate. So it does have an impact, and we're seeing it play out as we see more and more data. The other thing to point out when you think about biofertilizers and nitrogen fixers, phosphorus solubilizers, and then at the beginning I talked about the mycorrhizal fungi, we can see when you look at yield response from this meta-analysis, mycorrhizal fungi certainly have an impact, but more importantly, when we're looking at applying bacteria, when you apply a nitrogen fixer and a phosphorus solubilizer together, we're seeing a better impact than when they're applied separately. And there's reasons why. In order for nitrogen to be fixed, biological nitrogen to be fixed. It's a very energy-intensive process. And energy, you need phosphorus to drive that energy. And so having more available phosphorus there to fix nitrogen goes together. It's a biological system, okay? So some interesting insights from these meta-analyses. We could go on for hours and days, but we're not. One of the things I want to point out, we talked a lot about drought conditions, sandy conditions. This is our actual product that we brought to the market a couple years ago, BioYield, that contains a Pseudomonas species. And when we tested it, this is a species that was isolated from Wood County, so Northwest Ohio, cold, wet conditions. And what we found when we took it out and this was working with one of the major distributors in the US. They took our product and evaluated it all over the place. What they consistently found was that this product did very well in helping with an effect, a yield response effect, when it was cold or cold and wet. So we always saw a positive yield advantage effect under those conditions. When it was warm or dry, we didn't. Okay, so in this case, this microbe, because of where it came from and where it was comfortable being, we saw that impact directly on the plant. And so the other thing I just point out really quick is we told them not to use 1034.0 in this trial, and they did. And you can see 1034, 1034, and then we never saw an effect because it killed all the microbes, okay? So these are some of the challenges when you run field trials. If they're not listening to you, they do things that don't make sense. So we saw that in this actual trial, so for what it's worth. But again, be mindful of conditions. And so I'm going to jump over this pretty quickly, and then I want to get to the last slide. But summary of largest effects when we look at all those meta-analyses. For plant growth promotion, you see when there's more stressful conditions, particularly early season, that's when you're going to see the biggest impact of using these microbial products. It could be drought and heat, depending on the microbe. It could be cold and wet, just like I showed with our particular product. Sandier soils, higher soil organic matter, no-till, a lower yielding environment. These are areas where these plant growth promoting products are showing widely over geographies that they can provide a benefit, right? And so here are some of the strains that are currently in products that are well known. Back to biological nitrogen fixation. This was touched on earlier with soybean. If there's already a high rhizobia population, you're battling against what's there. But if it's low, this is where a soybean inoculant can really help. But also be mindful of what's there in terms of nitrogen. If there's a high level of nitrogen already there, these microbes they don't want to put the extra energy in. They're lazy. They're not going to fix nitrogen because it's already there. But if it's a lower amount, then you do see that effect of nitrogen fixation. And then if having soil available P, I showed that that combination of a nitrogen fixer and a phosphorus solubilizer can go together, right? If there's enough soil available P there, it helps with the energy that's needed for nitrogen fixation, okay? All right, best practices real quick. Make sure you understand the label and make sure there's a high concentration of CFUs to start, at least guaranteed by analysis. It doesn't guarantee they're there, but guaranteed by analysis it should be there. That drives your number of CFUs per seed. Store under appropriate conditions to prevent log loss, that's important. But then how you deliver it to minimize log loss is equally important. So seed treatment, use the same day as treatment if possible. I know you guys don't like that. The more days that go by, the more microbes you're going to lose. That's the reality of these living organisms, but keep that in mind. Dry application may be better in protecting the microbes from that dry down effect, so keep that in mind. In furrow, what you mix with matters, the starter fertilizer, all the way to the water you're using, okay? So keep that in mind. And consider new technologies like I showcased here. So where to start? Define what you want to achieve, right? If it's nitrogen fixation, focus on those products to start. If you're trying to mitigate certain stress, whether it's drought or cold wet, find products that'll help you do that, to help with that early vigor and early emergence. Understand what the product is and how it works. Go back to the label, understand from go to the manufacturer's website, whatever you got to do, talk to your crop consultants, but understand what it is. Consider your best zone, particularly if you have lower yielding areas. Try the product there first, right? We know that that's where you might see the biggest impact, so try it there. Consider your best application method. We went through quite a few, seed treatment and furrow, foliar. And then remember that not all value comes from yield, although that's what we're trying to achieve. You can have value, early vigor, helping improve your germination rate. There's things like that that are equally important, improving your ROI. And try at least 2 years. I would recommend longer than 2 years, but at least 2 years. Don't give up after the first year, right? We do see a lot of difference from year to year depending on what happens with the weather. So do it more than 1 year. All right, that is it. I don't have a snazzy QR code I would love feedback on this. Try to catch me here today because I continue to try to refine what the message, I guess, in using biologicals. So any feedback definitely helps me continue to improve. Questions? I know we ran.
Yeah, we got time for one question.
Very long.
What characteristics of that water?
Great question. Honestly, we don't know by the time but you have to wait for the microbes to grow. So by the time we didn't do the analysis on the water, to make a long story short, so I don't know. What I would imagine is that there were it was from a well source. I think there were some minerals in the water that just weren't compatible with the microbes, but I'm not sure. I wish we hadn't thrown that sample out before we made the measurement. pH, chlorine, it did come from a well, so I know chlorine's usually not as much of a problem from a well, but it's a good question. We were very surprised.